and just some background on thermoduric bacteria in general. So if you feel comfortable introducing yourself, if you want to go ahead and unmute, that would be fantastic. I'd love to know who we're having a conversation with today.
I'm Kathy Arnold from Twin Oaks Dairy in Truxton, New York. We've been organic for 25 years and ship milk to Upstate Niagara Cooperative. I've also been active in NOFA and NADPA, the Northeast Organic Dairy Producers Alliance.
Thanks so much for joining us, Kathy.
You're welcome. I wasn't sure I was going to make it, but
good to see you. Susan, I think you had your microphone turned on. Do you want to introduce yourself?
Yeah, I'm Susan Beal. I'm a holistic veterinarian who's particularly interested in grass-based agriculture, but that's not exclusively. And way back when, before my veterinary degree, I actually have a degree in ag bacteriology, ag micro.
Okay, fantastic. Thanks for joining us.
So I'm Rachel. I actually work in Nicole's lab, and Renee and I down at the bottom did a lot of the lab work for this project. So,
can you hear me?
Yeah, go ahead, Linda.
So I'm probably at the opposite end of the spectrum. I am late to farming. I started raising goats when I was 60, and I've been raising goats for 8 years now and milking them. And I just want to learn as much as I can about keeping the milk safe.
Cool, fantastic. Thanks for joining us, Linda.
I'm Jennifer. I live in Arlene, Montana, and I am just a small homestead dairy person with 2 milking goats. Been milking for the last couple of years, and like Linda, just want to learn a little bit more about keeping milk safe. I think maybe I might be in over my head with this particular Zoom meeting, just homestead wise. Thanks.
Sure. Yeah, welcome. Samantha, you want to go? Go ahead.
So I have a small grass-fed dairy farm in upstate New York. We ship to Organic Valley. I'm an educator with NOFA New York and do, run the helpline. So I'm really interested for my own farm and for farmers that I help.
Great. Wonderful. And then I think that the only person on my screen that I haven't seen is Mark. Mark, do you want to unmute and introduce yourself? If you don't feel comfortable, that's also just fine. Or if you wanted to throw something in the chat, that's also fine as well. Okay, well, let me share my screen and we'll go ahead and get started. I think, again, one of the benefits of having sort of a small group is we get to have a conversation. And, if you see something here that you have a question on that you just want to pop in the chat or raise your hand or something, we'll try to keep an eye on that. And, we can always stop and have that conversation. Okay, so can you see my screen? Looks good. Okay. So we're talking about thermoduric bacteria today, and the laboratory pasteurization count, which is one of the common quality parameters that a lot of dairy producers have assessed on their products. So first of all, though, let's just make sure we're all on the same page about what thermoduric bacteria are. So the sort of technical description is that they're bacteria that can survive exposure to heat treatments. I have a couple of different screens here, so if I'm looking in weird directions, I'm still paying attention to you. I'm just looking at my slides. So they can survive these higher heat treatments that that are considerably higher than the temperatures that they prefer to grow at. Most bacteria are fairly heat labile, meaning that they're killed by these higher temperatures. But thermoduric bacteria can actually survive vat pasteurization temperatures that are defined by the pasteurized milk ordinance, which is 63 degrees or 62.8 degrees Celsius or 145 degrees Fahrenheit for 30 minutes. It's pretty toasty, actually. It's quite warm, and these bacteria survive just fine. And so in order to enumerate or to count these particular bacteria in raw milk, we use the laboratory pasteurization count, which sort of mimics that pasteurization temperature time combination. So it's exactly the same, 145 degrees Fahrenheit for 30 minutes. So generally, there are two major groups of thermoduric bacteria that we find in raw milk. The first are bacteria that can survive that heat treatment because they form spores, and I'll talk a little bit more about what those are. And then bacteria that can survive that heat treatment without forming a spore, and they do this in just their normal vegetative bacterial form. So spore formers are an interesting group of bacteria. They're very sort of specialized at surviving under harsh conditions. And they do this through the formation of this really resistant structure that we call an endospore or a spore. Not to be confused with fungal spores. So fungi can often, like molds, can produce spores as well. Those are actually reproductive structures for fungi. Whereas in bacteria, they're not reproductive. They're simply there to provide resistance to harsh environments. And so I like this little diagram because, can you see my pointer when I move it? Okay. They're quite interesting because these bacteria can live in what we would consider sort of a normal vegetative state, where they're growing and multiplying and producing metabolic outputs. And then what happens is if they sense in their environment that there's some sort of condition that would be lethal to them, and that's typically things like reduced nutrients or reduced moisture, then they go through a pretty complex series of steps where they then package up all of their genetic material inside of this endospore. And the endospore is multiple layers of proteins, and it's essentially dehydrates itself. And then it becomes really, really resistant. And we know these spores can survive in natural environments for millennia. They're extraordinarily resistant. And then once that spore is in an environment where it can grow again, it will re-enter its normal vegetative cycle and start growing again. And so in dairy, you can imagine spores are present in soil and plant material and water. They're all over the place in the natural environment and in the dairy farm environment. And they enter into the raw milk. That raw milk then, would typically on a commercial scale go and be pasteurized or processed into a dairy product. If those spores are still in the spore form when they're in the raw milk, they survive that processing. And then in the finished product, in the fluid milk or the cheese or the sometimes like dairy reconstituted dairy powders, those kind of things, they can re-enter this cycle and then they can grow and cause defects in finished products, right? So they're kind of tricky from a microbial perspective in dairy products because we can't just apply heat to get rid of them because they survive that heat treatment. So that's the first group of thermoduric bacteria. They only survive in that spore form. If they happen to be in the milk in this vegetative form, that form is not resistant to heat, and so they will be killed just like most other bacteria. Some of the types that we find are Bacillus, Pennibacillus, Viridibacillus, and Leysinibacillus. You might recognize some of those names. And then the second group, again, these don't form spores. They just are heat resistant because of their inherent resistance to stresses. Some examples of those types of thermoduric bacteria are Microbacterium, Micrococcus, Brachybacterium, Cochlearia, and Streptococcus. So there's actually about 9 different genera that we know of that are commonly able to survive these heat treatments and that we would consider a thermoduric bacteria. So where do they come from in the dairy system? We actually know that they can enter into the system either at the farm, and that's what we're mostly concerned with today. But they do have in-plant sources as well that we also study. So just from a farm perspective, the thermoduric test or the laboratory pasteurization count has been used for 70 plus years in the US dairy industry. And it was developed as an indication of improper hygiene at the farm. So cleaning and sanitation of the milking equipment. The levels of these thermoduric bacteria could be quite high if there wasn't proper cleaning and sanitation. And so folks started using this test and applying it to raw milk, and it's been used ever since. There are some generally recommended guidelines for what is acceptable as a laboratory pasteurization count or a thermoduric count. Most folks say less than 100 is ideal. We want to have less than 100 colony forming units of these thermoduric bacteria per milliliter of raw milk. Somewhere between 1 and 200 colony forming units is, sort of like a high risk or sort of a medium problematic level, and then high is above 200 CFU per mil. And every cooperative or processor who uses this test has their own sort of limits for it and their own action levels for the laboratory pasteurization count. But these are sort of represent some generally accepted limits. We do know that because spore forming bacteria are found in this test, that the LPC count is often used as a proxy for spore count. And because spores can survive pasteurization, people are concerned about them because they can go on and cause issues in the finished product. And so a lot of folks have sort of used this as an indication that there are a high level of spores in the milk. And then just quickly, I'll briefly touch on the in-plant sources just so you get sort of the perspective on where they're coming from. Because these thermoduric bacteria are able to form biofilms, so they sort of set up shop in equipment that hasn't been cleaned and sanitized appropriately. And that's why we find them in inadequate cleaned and sanitized equipment at the farm too. The same thing can happen in the processing facility, especially in equipment that occurs before the pasteurization, because that equipment often doesn't receive as much attention as the equipment that happens after that kill step of pasteurization. These bacteria can often grow in this equipment until they reach pretty high levels, and then they can contaminate the milk as it flows through the equipment before it's pasteurized. And then because these organisms survive this pasteurization temperature better than most, then they can end up in the finished product. Most of these organisms that are not spore forming bacteria don't grow at refrigeration temperatures. So there's really a very select group of thermoduric bacteria that we see causing quality issues in finished product that is held at refrigeration temperature, which a lot of dairy products are. We do know, though, that there are some thermoduric bacteria that can show up in testing that's done on yogurt, for example. They don't grow under those conditions, but a lot of these products have specifications for bacterial levels that they have to abide by. And some thermoduric bacteria can show up on some of the indicator tests that are done to show whether there's been proper hygiene at the processing level. And so high levels can cause issues with conformance to those specifications. They can also, especially the spore forming bacteria, can cause quality issues in fluid milk and in cheese. And again, in specification issues in things like dairy powders. And of course, we're doing a lot of exporting of dairy products right now, highest levels historically in the US of powders and other products that are getting exported. And so processors are quite concerned with the level of these organisms in the powder because of the strict specifications for them if they want to export them. Okay, so I wanted to spend the rest of my time with you today talking about results from a study that we've recently been conducting in my program. And this has been really focused on spore-forming bacteria in the organic dairy system, so from raw milk through finished product. But as we were evaluating spores in organic raw milk, it became very obvious to us that generally thermodurics are an issue of concern to a lot of organic producers, mainly because these organisms are commonly tested for, again, for quality tests on the raw milk. And so we sort of designed a study to look into what are the types of thermoduric bacteria that we're finding here in organic raw milk, and what guidance can we offer to the industry. So I'm going to go through that. So we had a bunch of unanswered questions. There's not been a lot of research into thermoduric bacteria, either in organic milk or in conventional supplies. So there wasn't a whole lot of current research that had been done. There was a bunch in like the 50s and 60s, but I think you'll all agree that a lot of things have changed since then. We know milk quality has changed tremendously since then. So we felt like it was time to update what we knew about these organisms in raw milk. So some of the questions we had, what's the impact of temporary freezing on thermoduric bacterial concentrations and populations? And this really came up because sometimes when producers are subjected to this test, the test is used on their raw milk supplies. They want to be able to take a sample themselves and maybe have it tested at a different lab, or just to retain it in the freezer to make sure that if there's a problem, they have a backup sample that could be evaluated. But nobody has looked into what's the impact of, freezing up to 7 days on the LPC count, or the types of bacteria that are found after the laboratory pasteurization count. So that was the first question that we wanted to answer with this study. The second one, again, what are the primary bacteria types? What's the mix of spore formers and non-spore formers in contemporary organic milk supply, raw milk supplies? We also wanted to answer that question of, can we essentially use the LPC count as a proxy for spore count? If we have, for example, an elevated laboratory pasteurization count in a raw milk sample, does that tell us something about the specific population of bacteria that are causing that elevated count? So that was the second question we wanted to answer in this study. And then the third was, can we use MALDI-TOF MS, which is a tool that's commonly applied for mastitis organisms at the farm level? So, with bulk tank samples or individual cow samples, can we use that tool, which is more accessible to producers, to identify the thermoduric organisms in a raw milk sample? So to answer that first question, what's the impact of temporary freezing on thermoduric bacteria? We did a pilot study, and we collected 15 raw milk samples, and we essentially split the sample into 2 vials. One vial was stored at 4 degrees Celsius, which is just under 40 degrees Fahrenheit, until we did the laboratory pasteurization count. And the other vial was stored at a common freezer temperature, like a consumer freezer temperature, which is about -21 degrees Celsius or -6 degrees Fahrenheit. And we stored that for up to 7 days and then thawed and did the laboratory pasteurization count. And then we did a comparison of both the numbers that we got between those matched vials from the St. Bronze milk sample. And we also selected isolates, bacterial isolates from those 2 different matched pairs of samples and identified them using genetic DNA sequencing. And what we found, this is just a box plot of the total laboratory pasteurization count. And let me just walk you through how to look at this. So we have our fresh samples. Oops, sorry about that. The ones that were not frozen, so they're held at 4 degrees Celsius, and are frozen samples. And these are a box plot, what we call a box plot of the laboratory pasteurization count. And essentially what this is showing us is where is the middle of the data. So this is the median, the red bar is the mean, or essentially the average. What's the average? And then the spread of the data. So what's the highest level that we got way up here? What's the lowest level? These dots down here. And you can see these 2 box plots are very similar between the fresh and the frozen. And in fact, when we did the statistics to determine, can we say these are different or not, what we found is that there is no impact of the freezing on the LPC count. There was no difference between these 2 data sets. So that's good news. It gives us a lot more flexibility as an industry to take samples and freeze them and still be confident that the result is not going to be different. We're not killing off, we're not reducing the count, which is what the potential was to kill off bacteria with that freezing process. So then the second part of that is to look at what were the populations of thermoduric bacteria that we found in those samples, fresh and frozen. So again, we have our fresh samples here, we have our frozen samples here in this column, and the different colors just represent different genera of bacteria or different families of bacteria. And just with a quick look, you can tell that they look pretty similar, right? Most of the highest proportion were bacillus, which is a spore-forming bacteria. We have a lot of mycobacterium, which is this orange. We have quite a bit of brachybacterium. And in general, what we saw is that the numbers of these different types of organisms didn't vary significantly. And while we did find a couple of organisms in the frozen samples that we didn't find in the fresh samples, it was like 1 isolate of each. And so it was probably just by chance that we found it after the freezing and not in the fresh. So this gave us a lot of confidence also that we weren't, not only did we not change the overall level of thermoduric bacteria by freezing the samples for a short period of time. Again, we froze for up to 7 days. But we also didn't change the population of those thermoduric bacteria by freezing the samples. So we didn't, for example, lose an entire genera out of these samples by freezing them. So this is, really, really good news for folks who are using this test or are required to use this test for their quality programs. Okay, and then again, the second question that we had was, what are the organisms that we find in laboratory pasteurized organic milk? We just want to get a baseline. What are they? What are the levels? And again, is there an association between the level of those thermoduric bacteria in the raw milk and the type of organism that we find? Can we use the LPC as a proxy for a certain group of organisms? So as part of our project, we had 95 organic raw milk samples that we collected from across the US. They were frozen and shipped to us in my lab here in Ithaca. And we evaluated them for the laboratory pasteurization count, which again is 145 degrees Fahrenheit for 30 minutes. These are pour plated with standard methods agar. This is a traditional microbiological test. There's no easy way of, easy, quick way of doing this test, unfortunately, but it is pretty common. And those plates are incubated at 90 degrees Fahrenheit for 48 hours prior to enumeration or counting the individual colonies. And then from each of those agar plates, we selected a certain subset of the bacterial isolates, so the colonies that grow from those plates. We select them, we store them, and then we use typically what we do is DNA sequencing, because it's considered the gold standard for identification of bacteria, because we can actually look at a very high resolution at the genetic makeup of these organisms and evaluate what is it. So we did that, as well as that MALDI-TOF procedure, which I'm going to talk about a little bit more in just a couple slides. And we had 380 isolates that we collected for this project. And so just overall, as far as the level of thermoduric bacteria from the laboratory pasteurization count, again, let me just walk you through this plot so you see what we're showing here. So this is the laboratory pasteurization count. This is on log scale, which is the standard for representing microbial data. So here 0 is 1 colony forming unit per mil, 1 is 10 colony forming units, 2 is 100, 3 is 1,000. So you can kind of see how it increases by an order of magnitude for each of the log values that I'm representing here. And then just density as far as how many samples fell into each of these categories. And what you can see is that most of the samples fell in this mid-range here, less than 100 CFU per mil. And in fact, the mean or the average is 32 CFU per mil. So quite a bit below that recommendation of less than 100 CFU per mil that's sort of generally recognized as a good LPC count, which was really encouraging to us in this survey of organic raw milk. We did range from below detection. Our detection limit was as 5 colony forming units per mil, so quite low, to over 12,000 colony forming units per mil, which is of course quite a bit higher than what the recommended range is. So we had quite a spread of data, but most of the data fell right there in that sort of 50 to 100 CFU per mil. Okay, and then from each of those samples, those 95 samples that we collected, we selected bacterial isolates and did this DNA sequencing on them so that we could evaluate what are the types of organisms that we find. And overall, what we found, and again, let me just walk you through this plot so you know what we're looking at. Each of these bars represents an individual raw milk sample. And out of that raw milk sample, we categorized the bacteria that we isolated as either gram-positive spore former, so one of those spore-forming bacteria, a gram-positive non-spore former, so one of those other thermoduric bacteria, or gram-negative. Most gram-negative bacteria are not thermoduric, but we do have a couple that showed up in here, and they represent a very small proportion of the overall number of isolates, but we do have some in here. And then we had a couple that are represented in this purple color that we were not able to identify reliably with this technique. And so for each of these samples, we have, the color that represents the type of bacteria, and then the overall level of the LPC count as well. So for example, in this column here, you can see that we had some non-spore formers, some spore formers, and the count was 1.5 log. So each sample is represented that way from lowest to highest. And what we saw is that bacillus made up the most. So 40, almost 50% of the isolates that we collected were bacillus, and that was pretty much expected. And then we had a variety of other things: Brachybacterium, Cochuria, Streptococcus, Microbacterium, and then some other organisms. Overall, about 53% of the isolates that we collected from this study were spore-forming bacteria. And then the remaining were non-spore-forming bacteria. But within an individual sample from any given farm, the number of spore formers versus non-spore formers ranged from 0 to 100, meaning one raw milk sample may have no spore formers in it, and one raw milk sample may be completely spore formers. So that it was a huge range. And if we break this down by those generally accepted break points, so less than 100, 100 to 200, and greater than 200, what we saw is that in the less than 100 category, which most of our samples fell into, again, we had that 50-50 split between spore formers and non-spore forming bacteria, non-spore forming thermodurics. We had a few samples that were between 100 and 200, and a little bit of a higher proportion was spore formers there, but again, it was a very few number, very, very few number of samples. And then above 200, only 35% of the isolates were spore forming bacteria. We did look at this with statistics so that we could evaluate, is there a relationship between the level of LPC and the type of bacteria? And what we found is that there was no relationship, no statistically significant relationship, meaning that we can't use the LPC count as a proxy for a spore count because there is no relationship that exists between those two things, right? This leads us to believe that when producers are troubleshooting high LPC counts, they're not able to just say, okay, we have a high count, we can assume it's spore formers. That would make it really easy, right? If we could just be like, okay, well, if it's high, we assume it's spore formers, let's troubleshoot this in a way that we would for a spore former. And in reality, it can be high due to a spore former, or it could be high due to a non-spore former. And that's important because we would expect the non-spore forming thermodurics and the spore forming thermodurics to come from different locations on the farm. So what we really need to do is be able to say, what is the main type of thermoduric bacteria in a high LPC count sample, and then troubleshoot based on what the outcome of that identification was. And that leads us into that last question, which is, can MALDI-TOF be used as a rapid identification tool for these thermoduric bacteria? So MALDI-TOF MS stands for matrix assisted laser deabsorption ionization time of flight mass spectrometry, which is like, such a mouthful. So we don't say that. Let's just call it MALDI-TOF. But this is a tool that's increasingly being used, like I said, for identification of mastitis organisms. And so it is much more accessible in most cases than the gold standard, which is the other technique that I was talking about, which is DNA sequencing. So there are lots of labs that do that, but a lot of times it's, it's more expensive. It takes a lot longer time, and it's not as accessible to most people. So we wanted to be able to test this. Can we use this as an identification tool? One nice thing about this is once you have a agar plate like this that has the bacterial colonies on it, the time to identification using MALDI-TOF is less than an hour. So it's really quick. If we were to do the DNA sequencing, usually the fastest we can do that is like 3 days. So you can see that there's a huge difference in timing here as well. So we took all 380 of those isolates that we had collected from the samples, from the 95 organic raw milk samples, and we evaluated them and actually QMPS in Eastern New York was kind enough to do this for us. And they gave us the results of that identification. This is a table, and it's pretty busy, but it's a lot of information. So it's got two columns: the results from the MALDI-TOF and the results from the 16S DNA sequencing. And the way to interpret this table is, if you were to get a result of bacillus from the MALDI-TOF, how does that compare to what the isolates were identified as using the DNA sequencing? Okay, so 130 isolates were identified as bacillus according to MALDI-TOF. According to the DNA sequencing, 120 of them were bacillus, and the other 10 were a combination of other spore former genera. So close, but not exactly bacillus. Pennibacillus, MALDI-TOF said there were six of our isolates were pennibacillus. Our DNA sequencing said 5 of them were, and 1 was some other genera that belonged to the same family. So pretty close. Mostly the results were really similar, except for we had a huge chunk, almost a quarter of the samples that, or of the bacterial isolates that we had in the study, that were not able to be assigned identification using MALDI-TOF. So it's not necessarily that it provided a wrong identification. It just said it can't provide an identification at all based on the pattern that it produced. And we know that partially, at least, this is due to the database that's in these pieces of equipment, that the pattern from the analysis is compared against. So if there's no pattern that's similar to the isolate that we want to identify, then it just can't identify it because it doesn't exist in the database. So this is a solvable problem, right? If we want to be able to use MALDI-TOF to identify these thermoduric bacteria, we can update these databases to include this information that's compared to a gold standard like the DNA sequencing. And we can say these 48 isolates that were not able to be identified using MALDI-TOF, well, those are actually bacillus. We can upload those patterns into the database, and then now we have that there. And so with some kind of curation of those databases, this tool could be used as an identification tool for thermoduric bacteria, which will facilitate the ability of producers and cooperatives and processors who are using this test to do the kind of troubleshooting that's necessary at the farm level to reduce the thermoduric bacteria in those samples. Okay, so just an overview, we know that these thermoduric bacteria are present in the dairy system, both at the farm level and in the processing plant. They do have implications on finished product quality and conformance. And there are a number of producers who this test, the laboratory pasteurization count, is part of their quality program. I've talked to a lot of producers over the years, and especially over the last couple of years, who have expressed some amount of frustration with this test because they have a hard time with troubleshooting sometimes. And I think a lot of that goes back to the outcome of this study, which is that we really have to understand what are the organisms that are causing the problem so that we can do troubleshooting that's targeted toward those organisms instead of just general troubleshooting, which would most likely be focused on cleaning and sanitation of the equipment at the farm. So we have now an understanding of what these organisms are, how we can identify them, and what tools we can use for that. So I think that this gives us a really good understanding of moving forward, how to be more targeted in our approach to troubleshooting these laboratory pasteurization counts. Okay, so definitely happy to take some questions or comments. We can go back to slides if you want to. I do want to acknowledge, I know Rachel mentioned it as she introduced herself, but both she and Renee are on today, and they did the bulk of the experimental work for this project. So thank you both. And this is really wouldn't have been possible without them and without the funding from Organic Research and Extension Initiative. Any questions, any comments? Have folks dealt with troubleshooting for laboratory pasteurization count, or are you currently using that test?
So Nicole, I haven't dealt with a lot of LPC issues on my farm, but from a practical perspective, are there practical things that you would advise farmers to do to keep those numbers low?
Yeah, so there's a couple of things, and it typically comes back to hygiene in one way or another. So hygiene of the equipment, really, really important, because a lot of these organisms can, sort of adhere to the equipment and grow in the equipment. So then, when the milk flows through, it contaminates the milk. But also udder hygiene and end condition and hygiene can play a role. And we know that specifically for the spore forming bacteria that survive that treatment. So if teat ends are really rough or not adequately cleaned during the sort of pre-milking hygiene procedures, then those counts can be elevated because of that. So it really is just some general hygiene stuff, both at the cow level and the equipment level. So I know that Jessica has a few different resources she's going to send out to everybody. So that will have our contact information and some information on thermoduric bacteria, the recording from today. So, and you can always reach out to me if there's any questions that you have or you want to follow up for any information. Our goal is to provide a resource to folks when they need it. So feel free to reach out, and we're always happy to talk with people and interact. So feel free.
Thank you so much, Nicole. No one has any questions. Do you want to wrap up, or was there anything else that you wanted to go over?
Yeah, sounds great. If nobody has any questions, thanks.
Good. How's it going?
Do you have
Thanks so much, and thanks, Susan. We do have a paper coming out in the Journal of Dairy Science. It was just accepted with minor revisions. Wonderful work, Renee. And so it should be coming out in the next couple of months in the Journal of Dairy Science if you want to look at this topic more in depth in the results from this study in depth. I can throw the title into the chat for you so you can keep an eye out for it when in the Journal of Dairy Science. So let me just do that real quick. And we will have a number of other publications from this study that will come out over the next year or so, more focused on just the spore forming group of bacteria. And that's the title there in the chat that you can keep an eye out for it.
Nicole, is this going to be pretty science oriented, or is it something that we could put in a producer publication?
Great question, Kathy. So this one that's coming out in the Journal of Dairy Science will be very technical, but I would be happy to put together a shorter, more sort of generalist perspective on this paper as well.
Okay, I'll put you in touch with our main editor.
Wonderful, thank you.
Yeah, Nicole, we'll be happy to share that on our website as well.
Great, thank you.
Mm-hmm.
Okay, well, thanks everybody. Have a wonderful afternoon.
Thanks, you too.
All right,
Thank you.
bye-bye.
Bye.